Okay. All right, and everyone that is here, if you feel inclined to do so, you can, if you don't see the names of your breakout room or your Western blot writing team, you can go ahead and send them a message and ask them where they're at, because there are definitely people missing. And when that happens, then I will record some of this for participation points, so they will not get those points today, but you will. So we can go ahead and just review what we've kind of covered so far. So we know that we're going to be analyzing this protein called rubisco. And last week we kind of identified some of the key phrases that should be part of the research objective, in that we are looking at how expression levels of rubisco change, or can, maybe they don't even change, but we're looking at rubisco expression and the impacts that 60 minutes of light has on this rubisco expression in the model organism Pisum sativum. Okay, we're doing this using the technique called a Western blot. And the way that we're using it is more of a qualitative approach. We're just going to say we're going to be able to make conclusions about more or less. That's pretty much it, or equal amounts. We're not actually looking at concentration. So this is everything that we discussed last week. And so some of the predictions, we definitely talked about the controls. And so the positive control on this experiment is going to be plants that were grown in a 12-hour photo period, which means every day they get 12 hours of sunlight, not sunlight, 12 hours of light, and then 12 hours of darkness. And we chose this because this is going to kind of be the relatively high level of rubisco expression. And it does mimic real world growth conditions. And this is in contrast to the negative control. And these plants were grown in the complete absence of light for the duration of the growing period. And there is relatively low levels of rubisco, which is why we predict there should be low levels of rubisco. And then you and your breakout room or your writing team should have discussed what you predict the rubisco expression will be for the plants that were exposed to a 1 hour light duration. Right? 60 minutes, 1 hour, choose one and stick with it. So, I mean, the options are that these plants will have more. You always want to compare to the controls. So we've talked about this before. So we want to talk about the testable sample and we want to compare it to the control. And so since we have two controls here, we want to compare it to each control. So we can start with like, okay, well, one example is that the rubisco expression will be more than the negative control, but less than the positive control, right? So somewhere in the middle of that range. So you have the high, the low, and your testable sample somewhere in this range. Okay, that's one, and you have to give a rationale for this. So what is the rationale? Okay. So for that, you definitely are going to need some kind of reference. So maybe you have seen for other plants that photosynthetic enzymes, they are dependent on light exposure, maybe a linear relationship. So you definitely need to rationalize your prediction. Another option is that there's no expression. So it's very equal to that of the negative control. So we'll just say about equal to the negative control, which means it's much less than the positive control. And so you need rationale for that as well. So, you know, if you have found somewhere in the literature where they have tested maybe different photosynthetic enzymes or different plant models and showing that a short duration of light, like if you have withheld light for the beginning stages of growth, it doesn't matter how much light you shine at it at the plant unless it's been over, I don't know, a couple days of 12 hours of sunlight. I don't know, but that is an option. So you and your group have decided one of these options. And I guess the third option is possibly it's about equal to that of the positive control. If that's the case, then we know it's much more than the negative control. But the rationale could be that the plants have been deprived of light for so long that the second they're exposed to light, they are rapidly undergoing photosynthesis to the levels of that of plants that are grown in a normal 12-hour cycle. And you can keep going. There's definitely more options here, more possible outcomes. You might even argue that the plant goes into overdrive. So if it goes into overdrive photosynthesis, then it's going to make even more than the positive control. That's why it's important to have this range, but realize that you can definitely have samples, testable samples that may fall above the high end of the range. So we'll say that there are definitely more possible outcomes, okay? And so that's kind of where we're at. And we also covered last week the overall experimental methods. And so we talked in detail about this stuff here, the plant protein extraction and the Bradford assay, what we do with that information, why we have to do it, and so what it kind of the role that it plays in this whole entire report. And then we only briefly talked about the rest of these things. So the SDS-PAGE, that's the actual running of the gel, then transferring that to a nitrocellulose membrane, Ponceau staining that, and then immunoblotting with the antibody for Rubisco. So that's kind of where we're at now. And so last week we talked in detail about this stuff. This week we are going to talk about in detail the SDS-PAGE, the transfer, and the rest of the immunoblotting. All right, I think that's it, right? Yeah. Okay, so having said that, let's go ahead and, so we'll say in detail. We'll talk about the SDS-PAGE, transfer, and Ponceau, and the immunoblot. Okay. So we'll start with the SDS-PAGE. And so in the SDS-PAGE, as the notes from last week indicate, the reason why we put our plant samples in an SDS-PAGE or run them on a gel electrophoresis is to separate the proteins by molecular weight size. Okay. And so we also talked about how some of the reagents in the SDS-PAGE take us from quaternary and tertiary structure to all the folded up proteins, and we get them into their primary amino acid chains. And then not only does SDS help denature the proteins, but SDS also coats all the amino acid chains in a negative charge. And these two things combined is what allows proteins to separate by size as they're running through the gel. Okay, so at the end of the SDS-PAGE, you have a flimsy, clear looking gel with your molecular weight marker. Okay. Okay, and then you have a bunch of proteins that have also separated by size. So let me just make this a little bit bigger. Yeah. And you can't see these proteins at this point, but you can see the molecular weight marker, because the molecular weight marker, each of the protein bands is dyed with a certain color that is visible to the human eye. So you can see this, and you have proteins in this region of the gel separated by size. So in your first breakout room, these are the things that I'd like for you to discuss, and we'll come back and talk about them. So we're definitely going to talk about the methods in a bit more detail. Think about it like we're trying to make sure that your outline for your method section of the report is accurate. So this is kind of like the informal writing workshop for the SDS-PAGE section of the gel. So the methods, like you put them in chronological order, make sure that you have that. So you're going to be sent to a breakout room, potentially with people that you did not write your report with, but that's good. That's your chance to compare notes and come up with one collective idea of what you think is going to happen. Okay, and so let's see. I think today I will have you, I am going to assign a team captain for the breakout room. So you need to come up with a Google Doc. And on that Google Doc, I want the team captain to write everyone's names on that Google Doc. And then at the end of class, you're going to be turning that into Canvas. I'll set something up so that you can do that. So the methods in chronological order. And remember, you want to be as concise as you can. So the fewest amount of words, remember who your audience is. It's a scientist. So avoid things like labeling, avoid things like explaining how every single sample is prepared. Like if all the samples are prepared the same way, just bunch them together. Avoid describing how to make solutions or how solutions were made. Most importantly is going to be either final mass. Actually, more important is either final concentrations, or if you don't have final concentration, then you write the final mass. If you don't have the final mass, then you write the final volume. That's kind of the order of relevance or information. So one example is when you're talking about preparing the samples for SDS-PAGE, you don't need to tell me that you added 3 microliters of your protein sample and then 17 microliters of SDS dye. You can just say you for all samples, you use X milligrams of protein for a total volume of 25 microliters or whatever it was. And the remaining volume is the loading buffer. And then that tells the scientists everything that they need to know. Okay, so you'll do that. So in bullet points, right? There's more than 3 steps, I'm sure. And we'll talk about that and make sure that everyone has the exact details we need. And then I want you to start coming up with your predictions. Okay. So this has to be according to your hypothesis. So if you predict that there's going to be more or less rubisco for certain samples, what will be, actually at the SDS-PAGE, we're going to skip that part. We don't need that. Sorry, I'm going to rewrite the same stupid word. If the SDS-PAGE works, what do you expect? So this is just a statement. No need to consider at this point what would happen if the SDS-PAGE didn't work. We'll talk about that when we get to Ponceau. Okay, so if the SDS-PAGE works, what do you expect to happen on your dome, or what do you expect to have happened? And then that's it. That's it. So this first breakout room, we're just going to take it piece by piece. So we'll do SDS-PAGE. I think this first breakout room, we can do 10 minutes. 10 minutes, you're basically just coming up with a method section. So you should have already done this last night. You're just comparing notes and kind of filtering out things that you may not need. And I think that's enough time. So let's see. Okay, I think I'm going to keep these relatively small. Okay. And so there's going to be 2 or 3 people in your breakout room, and we'll just keep it easy. So whoever's first name is alphabetically first, you're the team captain for today. So make sure to start at Google, a completely different Google Doc with just you and your breakout roommates today, and we'll get going. So it's 10, let's call it 10:20 now. We'll be back at 10:30. Are there any questions before I send you to your rooms? Okay. Awesome. See you in 10. Okay, welcome back. So we are going to run this part of lab just a bit differently, just to make things run a bit smoother. So I'm going to actually ask the groups to unmute themselves. You don't need to share your screen, but you can just tell us what, when I'm asking you what the next step is, just unmute yourself. Whoever the team captain was, unmute yourself and tell us what you think the next step is. So we will start from the bottom. So let's do room 6. Whoever was your group rep, please unmute yourself. Tell us what the first step of doing the SDS page you think belongs in a lab report. Tell us what you think that is.
We put basically stating what SDS-PAGE stands for. And then we put that a 15.10 milliliter resolving gel solution composed of DH20 and 12% acrylamide mix and a 0.38 molar tris with a pH of 8.8 and a 0.1% SDS and 0.1% APS was used.
Okay, so essentially let's say, so I think what you're saying is that the subsection title should be the full name for SDS-PAGE, so full name, and then maybe you just had SDS-PAGE in parentheses.
Yes.
Okay, that shouldn't, yeah. So make sure that you're not saying SDS-PAGE stands for, that's not how we write a method section. So we're trying to build our method section for the lab report. So that's your subsection title, great. And then you said that the first thing that you stated that was done was that a 15.1 mil resolving gel was prepared or made, whatever. And then you had all of the ingredients in parentheses, the final concentration of all of those. Is that what you're saying?
Yes.
Okay, so we have final concentrations when possible. And for the final concentrations, they can be in mismatched units. Like if one of them is in millimolar, no problem. If another one is in percent, that's no problem. So we're not going to go over the math here because we have done various worksheets and actual in-class practice with how to solve concentrations. So at this point, we're not going to go over that anymore. I'll just see in the reports if it's correct or not. So yeah, I think that's great, right? That's all you need to tell the scientists is like, hey, we prepared a 15.1 resolving gel, and then in parentheses, you provide all the details they need. They can make whatever volume resolving gel they want because you gave them the final concentrations. That's great. So now, since we're on group 6 and we're talking about making the gel, so can you tell us what the next step is?
It was prepared and polymerized at 23 degrees Celsius for approximately 20-30 minutes.
Great. Perfect. Okay, so the gel polymerized, and then you provided the appropriate temperature and the appropriate time. Great. Perfect. This is awesome. Okay. Anything else about the resolving gel that you added?
I don't think so.
Okay, cool. Perfect. So let's move on to group 5. So what's next? Given what we just said about the resolving gel, this is going to be hopefully easy for you. What do you think should be written next?
Do we add a little bit of water to level out the polymerase?
So there's no polymerase in here. And adding the water is something that is not necessary to add in a lab report. So that might be something that you want to put in your lab notebook. But putting the water isn't going to be crucial for doing the Western blot. So what do you have next?
4.97 milliliter stacking gel.
Okay, what about it?
It consists of DH2O, 5% acrylamide mix, and then 0.13 molar tris, pH of 6.8, and then all of that. SDS and APS.
That's fine. So we're not going to go over the final concentrations, but you just said 4.97 stacking gel. What about the stacking gel?
We're gonna make it and then pour the gel.
Great. Okay, so 4.97 stacking gel was then prepared. You have all the information, ... And polymerized, and it's a template. So you just provide the the degrees that it polymerized at and the time it polymerized for. Great. So we're up to 2 sentences for the SDS-PAGE method section. And if you can imagine as we're going, so at this step, now we have this gel, right? So no one is talking about inserting a comb. No one is talking about putting together the apparatus. And that's good because we don't need to tell a scientist that. It's pretty self-explanatory. But what is necessary is that at this point, you have this region that you know is the resolving gel. It has already been polymerized. And then you went ahead and made the stacking gel. And then that's going to be kind of here in this region here. Okay, cool. So you got your 2 gels. Your gel is basically ready to go. At the end of this sentence here, if the stacking gel has polymerized, your gel is ready to be loaded and run. So let's go to 5, 6, 5, 4. Group 4. So what happens next? The gel's ready to go. So what do you do?
We set up the apparatus and run the gel.
So we just said we don't talk about setting up the apparatus.
Okay, well, then we prepare the sample buffer.
Okay, so you prepare the samples. How did you prepare the samples?
So the sample buffer is a mixture of 4% SDS, 5% mercaptoethanol,
Okay.
1% glycerol, 0.004% bromophenol blue, and then 12 millimoles of tris hydrochloric acid, which is pH 6.8.
Okay, so that's your loading buffer. So how do we prepare the samples?
We add 10 microliters of beta mercaptoethanol to 190 microliters sample buffer to make 200 microliters.
Okay, so you just explained how to make a solution. We don't want to do that. That is something that goes in our lab notebook, not something that goes in a lab report. So here's the thing, you already said what the sample buffer is. You said that it was a 5% BME in SDS loading dye or loading buffer, whatever you want to call it. Okay, you said that. So by then also saying that you added 10 microliters in whatever amount of other microliters, you are now telling the scientists, this is how we made the solution. They don't care how you made the solution. They are only interested in this. With that piece of information, they can make whatever volume they want to make. And so that is the kind of the thought process that we should be using when we write a methods section. You do not tell a scientist the volumes to add. They can figure it out themselves for sure. Okay, so you just said how to make the SDS loading buffer. That's fine. You still haven't told me how you're preparing your plant lysates. So how do you prepare the plant lysates?
Not sure.
Okay, who else was in your group?
I think it was Heidi and Megan.
Okay, Heidi, can you tell us how to prepare the plant lysates?
I can sure try.
Great, thank you.
Yeah, so is that the part where you add the buffer to the plant extract.
Exactly, you got it. So let's start with this. How much mass of each protein lysate do you think you need to add?
I'm seeing 25 microliters on here. Is that the right one?
That's the total volume. So let's write some of these important things down. So we have 25 microliters is the total volume that you're going to actually add to the gel. Okay. But you're not adding 25 microliters of each protein lysate. There's a specific mass of protein that you're going to add.
Okay. I don't see it on our notes, but I can try and track it down.
That's okay. If you don't see it on your notes, this is when we ask the rest of the class. So everyone open up your chats and tell me what you think the mass of protein is going to be added to each well. Great. Okay, thank you. So good job. I'm going to go up here and just remind everyone what we did so far and why that was important. So in the Bradford assay, we first needed to do the Bradford assay to get the concentration of all of your plant lysates. And the reason you need that is because you want to add the equal mass of each of these lysates, however many, if you have 3, if you have 10, if you have 1 million, you want to add an equal mass to each well of the Western blot. So coming down here to this little picture, here you want to add 30 micrograms of your positive control. You want to add 30 micrograms of total protein of your negative control, etc. So you want to keep doing that so that you don't bias and say like, oh, I'll just add 10 microliters. Well, what if one sample is way more concentrated than the other? Then you're starting with a different amount of starting protein, and that's bad. So each sample needs to be a total volume. So let's say this has to be 25 microliters. Okay. This consists of 30 micrograms of plant protein. And then the remaining volume, what is the remaining volume going to be? Well, back to Heidi. Or anyone from group 4.
Would the remaining volume be the 10 microliter sample buffer?
I don't think it's 10 microliters because it will be different amounts, but it is the loading buffer. So the remaining volume is going to be the SDS loading buffer. Okay. And so if you think about it, 30 micrograms of plant protein. So, oh, that's okay. 30 micrograms of plant protein. This is some of the calculations that I had you, I think, do, or you should have done. So it was definitely on the worksheet to kind of think about this. So let's do some fake numbers. So I think we're going to need to review this, given the information that has been shared so far. Let's say the positive control was 100 micrograms per microliter. Let's just say. And let's say the negative control was 32 micrograms per microliter. If you add 10 microliters, how much micrograms are you actually adding? So I'm going to give you a second to kind of think this out, and then we'll talk about it in a second. If you add 10 microliters, how many micrograms? So this is mass. So just take 2 minutes. 2 minutes to solve these 2 math problems. Okay, these are 2 different math problems. I want to know if you take 10 microliters of a 100 micrograms per microliter concentrated volume, how much mass of protein are you taking? And I want to know the same thing if the concentration of the solution was 32 micrograms per microliter. So I'll give you 2 minutes to do this. Probably not even that, probably just a minute. Okay, that should be enough time. So let's go ahead and start with the positive control. If you added 10 microliters of the positive control, how much mass are you going to actually be adding? Okay, so we've got a few people that are off by a factor of maybe 10 or 100. So let's review how to solve this. So if the concentration is 100 micrograms per 1 microliter, and if you are aliquoting or pipetting 10 microliters, and you need to solve for the mass, you take 100 times 10, which is 1,000. So crisscross multiply. So you take these two numbers and multiply them together, and then you divide by the remaining number. So 100 times 10 equals 1,000, divided by 1 equals 1,000. You know that your units are going to be micrograms. Okay, so you would be adding 1,000 micrograms, which is the same as 1 milligram. Fine. Now, go ahead and tell me, if you took 10 microliters of your negative control, how much mass would you be adding? So you'd be adding 320. You solve it the same exact way. So, great. Now, as you can see, if you took equal volumes, you would be starting with way different masses of total protein. That is not okay in a Western blot. You are looking at protein amount. So you can't say like, I'm going to actually start with positive control. I'm going to start with way more protein. Okay? So this is the reason why we needed to calculate the concentrations using the Bradford assay. And then from there we say, okay, well, I should add an equal amount of mass of all of my samples. I told you 30 micrograms, okay, that's maybe a standard Western blot amount. So then you say like, okay, well, I don't want to use equal volumes. I want to use equal mass. And so with equal mass, you would solve, let's do it. So we're still going to use these same concentrations. So now I want you to do the math. And the question is, how much volume do you need to get, let's call it 50 micrograms of each sample. Okay, so the question is like, what do you set your pipette to for the positive control so that you get 50 micrograms? What do you set your pipette to for the negative control to get 50 micrograms? So I'll give you another minute to solve this. Okay, that should be enough time for you. So for the positive control, how much volume would you need to acquire 100 micrograms? Thank you to those who entered their answer. So it looks like most of you are getting 0.5 microliters. And so this is just another ratio that you set up. And so if your goal is to get, what did I say, 50 micrograms, the question is, how much volume do you need? So again, you do crisscross multiplication. So you take the numbers diagonal from each other that are there. So 50 times 1. And then you divide it by the remaining number, which is 100. So it's 50 divided by 100. This gives you 0.5 microliters. So you'd set your P2 to 0.5 microliters, and you would take out 0.5 microliters. That would be 50 micrograms. Okay, how about for the negative control? You solve it the same way. So you take these numbers diagonally across from one another. So 50 times 1. And then you divide by the remaining number, which is 32. And this is going to give you 1.56 microliters. So I hope that you can appreciate that these are different volumes, but they're the same mass. And that's what you want in the Western blot. So this is same mass of each sample. When you take the same mass, but the samples have different concentrations, essentially you're going to be taking different volumes. Same mass, but different concentrations means different volumes. And so that's why I wrote that up here, that if you are trying to take 30 micrograms of your plant protein sample, if you had 10 samples or 15 samples, you're likely going to be taking a different volume from each plant sample. That's why the remaining volume is going to also differ. So let's say that was the case. Let's say we did want 25 microliters. We've calculated that we only need 0.5 microliters of your positive control. So this is a very easy one for everyone, I hope. What is the amount of SDS loading buffer? If the total volume needs to be 25 microliters, what is the amount of loading buffer that you're going to need? Yeah, 24.5, exactly. So if you need 25 total, 0.5 of it is from actual plant protein, The rest of it is SDS loading buffer, yeah, 24.5. So the remaining volume is also going to be different volumes per sample. So in the positive control, for example, this in our example, not in the actual experiment that we did, it would be 0.5 microliters of protein, 24.5 microliters of SDS-PAGE, and then we have a total volume of 25 microliters. You would do that for each sample, so that each sample, when you load it into your well, not only do you have the same mass of total protein, you also have the same total volume, so 25 microliters. Okay, any questions on this? Okay, so yes, Megan.
Would we have to include, we don't need to include like any of the like way we're solving this. I don't know how to verbalize this. We don't need to include that we're making it equal masses in our methods.
No, okay, so that's where I'm going next. So the what I was gonna say exactly next is like, okay, so what do you put into the method section? And so here's what I think needs to go into the method section. Let me move all this junk around. This is last week, so let me just get it out of here. Okay. Right. So we'll go back to, everyone, we'll go back to black so that we know that these are the important things that go in our report, okay? All the colored stuff is just information for kind of understanding the math a little bit better. So what goes in the report about prepping the samples? Here are the important details that we definitely need. All of your samples, I don't care if you had 5 or 1 million, you would be using the same mass. So you would say, and in this case, everyone told me it was 30 micrograms of each protein lysate was used, right? The other important thing, like what are the minimum details you would need to tell a scientist? The scientist would also need to know that the total volume was of each sample was 25 microliters. And then you have to tell them that you're going to add SDS loading buffer. So then you say the remaining volume is SDS loading buffer. And then here's where you put all the information, the 5% BME, whatever else was on the recipe worksheet. Okay. Samples, Western blot samples were prepared with 30 micrograms of each protein lysate used for a total volume sample of 25 microliters, comma, or semicolon, or new sentence. The remaining volume is SDS loading buffer, all the ingredient final concentrations in parentheses, period. So this could be literally 1 to 2 sentences. Okay, questions on that? And that did answer your question, right, Megan?
Yes, thank you.
Okay, awesome, great. So let's review so far what we have. We have how the gel was prepared. So a resolving gel, a stacking gel. And so I'm gonna actually erase this little part right here because we've already said it in the sentences below, right? So here is the samples are prepared. So now if you think about it, we go back to our little cartoon, we have 3 new tiny little microcentrifuge tubes. They each have 25 microliters. They each have some amount of loading dye, so it's blue, okay? Some amount of protein lysates. These right here are ready to be put into the gel, and the gel's ready to go. So we stopped with group 4, so let's go with group 3. once the gel has been prepared, once the samples have been prepared, what happens next?
So you're going to load the samples into the gel, and then you can set the gel, you're going to run it. So you'll run it at 150 volts, and we ran it for about 70 minutes.
Gel was run at 150 volts for 70 minutes. If it's approximate, you write approximately 70 minutes. At what temperature?
Room temperature.
Okay. Okay, so make sure that we have that there. And then this is kind of similar to the TLC report. So is 70 minutes the magic number? Is everyone runs their Western blots in the whole world, every single Western blot for 70 minutes, or what are they really looking for?
You just want to make sure that it doesn't go over the gel, but it like runs to like the top of the gel.
Not the top of the gel, because he started at the top
Or
of the
the
gel.
bottom of the gel.
Yeah, and so if you had to come up with kind of like, you know, for the TLC, we said that the samples would run until they were about, I don't remember the number, but maybe like a centimeter, well, not a centimeter, a millimeter, a centimeter from the top of the sheet. So, okay, so Kiana, thank you. So about 3/4 of the way through, and the way that you can tell that is you'll see the loading buffer. That's why there's bromophenol blue, so that you can see when to shut off the power. So at the end of this, what we're saying is you want to stop it until you see the blue line approximately 3/4 of the way down the gel. And I think that that is an important detail to add into the material methods until the loading buffer is about 3/4 of the way. Just keep in mind, we're writing notes right now. You should be writing full words in your method section. Great. That's it. And so if we look at our little picture, if this is where we're at, the gel's done. We're done. Okay. And so really, you have, I'll make nice big bullet points. This is probably a sentence. This is probably a sentence. This is 1 to 2 sentences. We'll put 2 bullet points there. And then here's one more bullet point. Oh, we're missing one detail. So what also needs to go in the gel besides your samples? This is for everybody.
A ladder.
So what other thing? Yeah, boiling. Oh my goodness, yeah. We have not actually talked about the samples weren't done being prepared. Thank you. Okay. So, Arlene, can you expand on the boiling part? Because that's important. You have to say that. So not only did you make these little samples here in your little tiny microcentrifuge tubes, but then you boiled those samples. So just give me the details about the boiling.
Yes, they had to be denatured for 5 minutes, and I guess you would say, would you say 100 degrees Celsius, or would you just say boiling water?
I think that if you took an actual reading of the temperature, you can say that. But if it's boiling, you can assume it's 100. So I think either way is fine. So samples were, you can just even say boiled or denatured in boiling water.
Okay.
For how long?
5 minutes.
Okay. Great. Now the samples can be put into the gel. So this is one more bullet point here. Okay. Great. So we got them. So this is a pretty short paragraph for your material method section, the subsection title, sodium dodecyl sulfate dash polyacrylamide gel electrophoresis, in parentheses SDS-PAGE. We talk about each of the gels being made and their final concentrations, and then polymerized for X number of minutes at X degrees Celsius. And then to prepare samples, we already talked about how we would say that. Samples were then boiled, or samples were denatured in boiling water for blah, blah, blah minutes. Samples were loaded into a gel, and then here's where we need to actually add the other thing that was loaded into the gel. And some of you already told me what that was, which is great. Let's see, what did you tell me? Yeah, the protein size standard, Bio103. So you have to come up with the actual product name, because that's not the product name. So you would, yeah, Leilani, nice job. Okay, so Bio103, you would start with precision plus protein kaleidoscope molecular rate markers, and then in parentheses, Bio103 161035. We're loaded into the gel. Now, if there was a different volume than 25 microliters, you want to state that volume when you're talking about loading the samples into the gel. So that's something that you got to go back to the video and check out and make sure, like, did Dr. B actually put all 25 microliters in there, or did she say that she put only 20, or what is the deal? If it's still 25 microliters, no need to say all 25 microliters were added, but if it's a different volume, you want to say that volume. So maybe you're going to add something about the volume of samples were loaded onto the gel, but you also need to say the volume of the marker. So it's the official product name, whatever the product name is. Okay. And then in parentheses, you just say Bio103 and then whatever the catalog number is. And we're loaded. Okay. Into wells of the gel. Okay. The gel was then run at 150 volts for about 70 minutes at whatever temperature until the loading buffer was about 3/4 of the way down the gel. Great. Let's move that over. Okay. Okay, let's get rid of this junk. Okay, cool. And now you can see that the gel's done running. Okay, any questions on the SDS page method section? Great, so we'll stay with group 4 and you're gonna finish this off here. Wait, yeah. What do you predict? So if the SDS page works properly, what should happen? What should have happened by the end of the SDS page gel? Group 4.
We predicted that if the SDS page worked, that we'd expect to see different bands separated by molecular weight for each sample.
How will you expect to see them when proteins are invisible?
We used a dye.
We haven't used a dye yet.
Would you be using, would you have to stain it first to see if it worked with the, the Ponceau red?
Yeah, so I'm just asking at the end of this, what will it look like? So you're right, you do have to stain it, but we haven't stained it yet. So at the end of running a gel, what will the gel look like? Nothing. Natalie, yes, thank you. You will not see anything. The only thing you will see is what?
You'll
The
see
ladder.
The ladder, yes. And this is why I want to have this conversation, because I really want students to really think about what they would see. And you're gonna see it because actually you should have already seen it because it's in the video. So at the end of the gel, it's a clear see-through gel about this big. The only thing that you see on there is the molecular weight marker that has separated by size. So seeing them as individual bands on the length, or I guess the height of the gel, tells you that separation did happen. Not only that, but you will also see, if you stopped it in about, when the sample dye has reached about 3/4 of the way down, you will also see a blue line across. That's all you will see. That's it. Okay. But if you do see the bands of the marker, the molecular weight marker, if they're nice and separated, then yeah, you expect The only thing that you can expect to see here is that the molecular weight marker bands have separated. Okay. By size, the smaller ones on the bottom, the larger ones on the top. And if that's the case, then your SDS page, at least with the evidence that you see. So what is the evidence? So this is what you expect. The evidence would be colored bands from top to bottom, right? That would look something like this, right? There's a couple different colors in there. That's it. That's all you would expect to see. That would be evidence that the SDS page did in fact separate the proteins by size. You still have no clue what's happening with your samples. Okay, any questions on that? Okay, great. So we can move on. So I don't know whose voice it was because the screen didn't pop up, but somebody shared that in order to see anything, you would have to stain it. But we don't stain yet. So everyone open the chat and tell me, what is the next step? So after you run this gel, we've already talked about it before, so it's not like a huge surprise. After you do your SDS page, what do? Please enter it. You transfer to the nitrocellulose membrane. And we talked about why we transfer it to the nitrocellulose membrane. Why? Because the next step is going to include immunoblotting, and you cannot immunoblot on a gel. Why? Because the proteins are inside of the gel, not on the surface of the gel. So when you transfer to the nitrocellulose membrane, you are going to immobilize the proteins on the surface of the gel. On the surface of the membrane, which is what you want. So, and we also talked last week, why do we use nitrocellulose? We use nitrocellulose for two really important reasons. Here are the properties of nitrocellulose that allow transfer to even work. Yes, so Christian's adding that it has a high affinity to proteins, absolutely. So the surface of it, if you're trying to get proteins on the surface of my hand, hopefully my hand's nice and sticky and it will stick to the proteins, but you can see that unless you took it, I don't know, like a drill and drilled through my hand, nothing's going to get through my hand. It's going to stick on the surface of my hand. Nothing's going to go through my hand because my hand's impermeable. Okay. So high affinity, impermeable. So now you definitely have your nitrocellulose membrane with your proteins on top, accessible to any antibodies that you're going to add to them. This is what you want. Okay. So the next step is, remember, in material methods, you don't need transition sentences in your results, in your discussion. You do. So one of the, an idea for a transition, if you're talking about results or something like that, is after the Bradford assay, proteins were run on an SDS page, then transferred to nitrocellulose. After they were transferred to nitrocellulose, they were stained with Ponceau. And now you can start talking about your Ponceau data. Okay, we're not talking about data right now. We're talking about methods. So after you write your SDS page method section, you just start a new subsection titled transfer to nitrocellulose and just start talking about that. Okay, so we're gonna do the same thing, method section. So this gets you back in the habit of what we're looking for when we come back to our group discussions. Have your bullet points really specific. In past tense, you're not giving directions, you're saying what happened. And you're not saying who did it, you're saying somehow it magically happened, right? So put it in the context, in the verb tense that should show up in your method section in your report, just like we did here. A 15.1 resolving gel was prepared, not Dr. B made this, or here's how you would make it. First you add, we're writing the method section. So let's do the same thing for transferring the gel. So transfer to nitrocellulose membrane. Include any important product names, vendor names, catalog numbers, always your concentration if you have, final concentrations if you have that, final volumes of like the whole entire thing, not individual volumes of reagents. Okay. And we'll make a quick list of the important details that need to go in the method section of this part. So another 10 minutes in your breakout rooms. Now, if you were in a group of 3, the next person that goes alphabetically, you're now the team leader. So keep adding to your same document, but go ahead. So the things that we're talking about in class, they should be on your document as well. And go ahead and add to that. So we're only looking for the methods in chronological order that a scientist will need to know. And at the end, what do you predict you will see if everything goes right? Okay. Just be able to describe that. Questions on what you need to do in this next breakout room? Okay. So alrighty, let's go ahead and get started. So we were talking about the transferring to nitrocellulose membrane, and we are putting together some notes for a good, concise, and succinct method section for that. So let's go ahead and start with breakout room number 3. If you can unmute yourself and tell us what the first step to doing this was.
So the first method is to remove the gel from the apparatus and remove it from the glass and submerge it in transfer buffer and place that on a rotary shaker at 50 RPMs for 10 minutes. And you're also going to place the foam, filter paper, and membrane all in the transfer buffer as well.
Okay, so gel was removed from the Bio103 Western blot apparatus, and saying that it needs to be removed from between the glass plates. That's part of removing it from the apparatus. So we'll just keep it really concise and say the gel was removed from the Bio103 Western blot apparatus and incubated in transfer buffer. And you don't have to tell me the recipe, but I'm going to assume that you have all the details of the final volumes of whatever makes up a transfer buffer in parentheses, right?
Yes.
Okay, good. For X number of minutes at X degrees Celsius on a rotary shaker at some RPM. Perfect. Okay, and then you also said, so that's kind of like you're imagining the gels in this dish and it's kind of rotating on the RPMs and some kind of liquidy buffer, great. Not only did you have to incubate and equilibrate the gel in transfer buffer, you also had to do that for all of the ingredients or all of the reagents that are needed for making the blot sandwich, correct? Mm-hmm. And so I think you can just say, so can you just maybe list them? I'm trying to think what would be the best way to do this. Okay, I know. We can say something like like once the gel is equilibrated, the end result is basically like you equilibrate the gel, but what you're trying to make is a blot sandwich. Mm-hmm. Okay, so you're just saying that a blot sandwich is was prepared. Mm-hmm. And then I think that the best way to do this so that you don't have to say the same sentence 500 bajillion times is say, was prepared using the following reagents in this order, and then just list them, right? Because all of them, all of the, like the foam pad, the filter, the membrane, all of those things had to equilibrate and transfer buffer as well. And so I think you can say something along those lines that all reagents were also equilibrated. in transfer buffer. And if there was just a minimum time for a minimum of 5 minutes each, or so something like this, some kind of approximate time. And so if you're imagining this happening in the lab, so the gel has been rotating on the shaker in some liquid buffer, and then you collect some sort of reagents that you're going to tell the scientist that's reading your paper in just a second what those reagents were. They're also going to be equilibrating in transfer buffer. If they were also on the rotary shaker, I don't think they were, but if they were, you would say also at our 50 RPM. If they weren't, then you don't have to say, but we're not shaken. You just don't mention the things that you didn't do. Okay, and maybe you just say, was prepared in the following order. And then you just make a list of each thing, one by one. So right here, there's a lot of different ways that you can kind of rearrange these details. This I would imagine to be probably 1 to 2 sentences, where this is just going to be 1 sentence for sure. And you and your writing team can figure out how to do that. But I think that these are the only important things here. That's kind of like preparing everything to get it going for transfer. So thank you to group 3, that was perfect. Yeah, Greg.
Yeah, quick question. So I remember in one of the videos, one of the rotary shakers broke.
Okay.
Was it this one, or do we need to make note of that in our report?
I don't remember if it was this one, honestly. It might have been during the immunoblot phase. So I will go back to the video and check it out, because I don't think it was this one. I don't even know if here's what I'll say about that. If it is obvious how long the reagent was on the rotary shaker for, then you should report that. If it's an instance where, the shaker wasn't working at all, then you don't say the things were shaken. And you don't say, we were supposed to shake these things, but the rotary shaker, that's like explaining stuff in the methods. You just don't say that you shook them at any RPMs. You just say incubated at blah, blah, blah time. Does that make sense?
Yeah, thank you.
Yeah. So if it was this one, I'll highlight it. So Avery is saying that it did happen here. So if the gel was not shaken or rotated, it's not shaken. It's really like this rotation like this. If it wasn't, then you don't include this part. If it was, put it in. Okay. Shaken. Yeah, shaken, not stirred. Okay, great. So at the end of this, just try to envision what you have. So you have, your foam pad, you have a filter paper, you have your gel. So whatever the correct order is, you have another, the membrane, another filter paper, and then another foam pad. Here's your delicious sandwich. Okay. All right, you're done. So you've created the blot sandwich. So what happens to that sandwich is what we're going to talk about next. So that brings us to group 2. So group 2, team leader, go ahead and unmute yourself and tell us, what do you do with this, or what happened to this blot sandwich?
So we placed the blot sandwich oriented a certain way. I'm forgetting, 1 second. So the nitrocellulose membrane is placed towards the red node.
Okay, give me 1 second. So the blot sandwich was placed into transfer apparatus. Yeah. With, you said, the gel facing the red node?
Yes.
Okay. That's the positive node, right? Yeah. Red, bunch of red, positive, yeah. Okay, and then what?
And then the nitrocellulose portion's placed towards the negative node.
Yeah, so I
Right.
think that would probably be a redundant 2 sentences, because if the gel is closest to the red node, then you know that what's on the other side is going to be closer to the black node. So pick one of them and just say that. So you can either
Gotcha.
say with the gel closest to the positive node, or you can say with the nitrocellulose membrane closest to the negative node. Either way.
Okay, gotcha. So then,
But I'm thinking that that actually should be opposite, right? You should have the nitrocellulose closest to the positive node so that the proteins can run toward it. Right?
yes.
Okay. And then, so once that's in there, once that's in the transfer apparatus, what do you do next?
Next, you turn on the power supply.
Nothing else goes in the gel box?
We put in transfer buffer. Oof,
How much?
I'm forgetting. Let's see. I don't remember. Is it 350 milliliters? That's good.
That sounds like maybe a little too lit. It's not enough. Okay, Kiana says 1 to 1.5 mils. Mils. That's like this much liquid.
Wasn't it just covered?
20 mils, so I have a bunch of things. So what was covered?
The membrane. Oh, I mean, sorry, all of the apparatus.
Yeah, so it looked like that. So you put this in a big tank. Well, this is like in some apparatus. You can't see it anymore. So it looks kind of like this. You have this big tank and some amount of transfer buffer was added. And so if the video doesn't say how much, it's definitely not 1 milliliter. 1 milliliter is like not even a shot. You guys know what a shot is. 20 mils is like maybe a tiny bottle of water. So yeah, there is a line on the tank that says transfer. And it's probably about 1 liter. So 1 liter is like a big Nalgene. Okay. So we can just call it 1 liter. But yes, Arlene, you're right. It's you fill it up until, sorry, let the train go by, so I'm not competing. Okay, so you pour enough transfer buffer until the transfer apparatus is fully submerged. Okay, so we'll add that as a detail. Okay, so now you have your transfer buffer in here too. Okay, it's pretty self-explanatory that you put the lid on with the little nodes. Okay, so we imagine the lid is on. All right, so Greg, go ahead and tell me the rest of what needed to happen here. So now we have the tank, we have the blot inside, we have the transfer buffer, the lid is on. So then what happens?
So yeah, then you turn on the power supply and you turn it on at 350 milliamps.
Okay, so the transfer was run or conducted, performed. We don't need to say we turn on the power supply. We're going to say the transfer ran at 350 milliamps for how long?
For 1 hour.
At what temperature?
At room temperature.
Okay. Always concentration, always when we have it. Or if we're dealing with electricity, always some kind of units of electricity, and then time, and then temperature. And we're good. Okay, that's it. Now, thank you. So let's go ahead and at the end of this transfer, I would like, Craig, you were group 2. Is that where we're at right now?
Yeah.
Okay, so group 1, team member, team captain, tell me what you predict to see. Like describe what it is, describe if it's a gel, describe what you think you're gonna see once you take all the lid off and you take the blot sandwich apart. What do you expect to see if this worked correctly?
What we expect to see is that the molecular ladder would be transferred over to the nitrocellulose.
Okay.
There would be nothing left on the gel, showing that it's a successful transfer.
Great. Cool, that was perfect. So yeah, so you have your nitrocellulose membrane. It's this tiny white thin piece of flimsy paper, and we are expecting that same banding pattern that we saw on the gel. We are expecting to see that on this flimsy paper, right? So there it is. You do not expect to see the blue line. That should not transfer over because that blue line is dye, it's not protein. And the rest of that should be completely clear and white. And then if you looked at the actual gel itself, so this is the nitrocellulose membrane, and if you looked at the gel, this would be clear because the proteins from the latter transfer to the nitrocellulose membrane. And what you're predicting is actually happening here is that there's no more proteins in the gel. And you're expecting here that the proteins, you can't see them yet because they're still invisible, are on the surface of the nitrocellulose membrane. Okay, so if you looked on this, if you looked at a side view, you would have all these proteins all along the sticking out the top of this nitrocellulose membrane, kind of like spines of a cactus, just sticking out. And if you moved this and looked at it in the side view, like you're looking through it, there's going to be nothing in there anymore. They have successfully transferred. Perfect. Thank you. Who was that? Was that Christian?
That was me.
Thank you. Great. Okay. So that's it. That's your transfer to nitrocellulose membrane. So I think it's a good time to go ahead and take a break. And when we come back, we're going to do the same thing for the Ponceau and for the immunoblot, and then we'll call it a day. So let's be back. Hopefully 12 minutes is good. Let's just be back right at noon and we'll get going. Okay. Questions before we take a break? That was really good. So thank you to the teams that contributed. And I think now we're in a good flow of what's expected. So I will see you at noon. Okay, so hopefully everybody had some time to take a quick break, grab a snack, do whatever. I had planned to do that laundry back there and fold it, but I didn't. I painted my nails instead. So let's get going on the second to last section that we're going to discuss today. And that is Ponceau staining. So you're going to go to your breakout room again, and you are going to talk about the method in the same manner that we've been doing. So what are the steps that were performed in succinct but specific details? And then in the prediction part, so this part's going to take you a little bit longer. So you're going to have 15 minutes for this because the Ponceau staining is super quick. For the majority of your breakout room, you're going to focus on predictions. Oh no, this thing's not working. Give me a second. Come on. Okay. Cool. Ah, busted. Okay, so predictions. Come on. Okay. We're good? We're good. So I would like for you to do a couple things with your predictions. So first and foremost, obviously predict what you think you're going to see if everything with the Ponceau staining worked as it should, and everything so far has worked as it should. So the SDS-PAGE worked, the transfer worked, the Ponceau stains the way it's supposed to stain. But when that happens, you should also incorporate if your hypothesis was correct. So if your hypothesis is correct, I'm pretty sure most people, and it doesn't have to be this, but where'd it go? If your hypothesis is correct, what would the Ponceau stain look like? Most groups predict this. If you didn't, it's not wrong because, well, I'm not going to say it, but there are different predictions. If you predicted this, what would your Ponceau staining look like if the 60 minutes of light actually made the plants produced more Rubisco than the negative control, but less rubisco than the positive control. What would the Ponceau stain look like? And when we're talking about the Ponceau stain, and we're talking about what would it look like, I put this little star to kind of remind you, when you're talking about it, if you're the group that's sharing it with the class, you're going to talk about, okay, well, you know, you might describe, we talked about last week how Ponceau is going to be red. Okay, so you know you're going to start with a description about red staining. Is it polka dots? Is it bands, stripes? What is it? So what do you expect to see? Do you expect to see bands that are thicker at some size or not? Thinner at some sizes or not? These details right here is going to be how you explain how your results would show that your hypothesis is correct. So what is the evidence that you're going to see? So here we're talking about actual evidence. So what is the evidence that you would see that would indicate that your hypothesis was correct? What is the evidence that you would describe that would show you that the Ponceau staining worked for all the samples the way that it should? Okay, that's one prediction that you have to come up with. A completely separate prediction is, what would the Ponceau stain look like? What would be the evidence that would show you, oh man, looks like I forgot to add a sample? What would that look like to you on your nitrocellulose membrane? So keep in mind, we're just taking this here and we're staining it. Okay. So what would that thing look like? So basically you're going to have this 3 different times, right? 2, 3. Okay. So this would be if everything works the way that you should and your hypothesis was correct. This would be evidence. What would it look like if you forgot to add a sample, but the other samples are totally normal? And then this, what would the Ponceau stain look like? What would be the evidence to show you that the samples were not fully denatured? So essentially, these are the conclusions. And this over here would be the evidence. So what would be the evidence so that it would allow you to make these separate conclusions? Okay, so that's your prediction. So let's move these methods over, because these are not methods that I just drew a picture of. 5-6 minutes on methods, 7 to 10 minutes on your prediction. So let's actually give you 18 minutes. So it's 12:07 now. Let's give you 18 minutes. That puts us at 12:25. Are there any questions on what needs to be talked about or discussed at your breakout rooms right now? Okay, so you should be adding to your breakout room notes. If you have Notability or Access like that, you can draw, or you can simply just type and explain what you think the predictions would look like. Okay, great. So we'll see you back at 12:25. Okay, so we can continue our conversation. And we're going to go ahead and start at the top this time. So we're going to start with breakout room number 1. And breakout room number 1 is going to share with us what they think the first step in staining your nitrocellulose with Ponceau is. So group 1, go ahead and mute yourself and let's hear it.
Okay, so for the first step, we have 0.1% Ponceau is added to 1% acetic acid.
Oh, so you're describing how the Ponceau stain was prepared?
Yeah.
Okay, not needed. You can just say whatever 0.1% Ponceau in acetic acid. Mm-hmm.
Okay, so then 20 milliliters of the Ponceau stain was poured into a weigh boat in order to fully cover each of the 3 blots, and then incubated for 5 minutes while on the rotary shaker for 50 RPM at 22 degrees Celsius.
Great. So there may have been 3 blots, but each blot is for a different lab section. So you're going to assume, you're going to write as though it only happened for one blot.
Got it.
Okay. Yeah. Okay. So we can say that the blot or the membrane, I think most people just use the word blot. So the blot was incubated with whatever the correct Ponceau is. I forget what the percentage is off the top of my head. So it's something percent Ponceau red in something percent acetic acid. And then the details that you told me that were important was the time, the temperature, and the RPM, and that it happened on a rotary shaker. Oh, wait. Volume. Great. Perfect. Okay. Awesome. Thank you. So go ahead and why don't you give us one more step of the method. So after you have shaken your little membrane in the Ponceau red for X number of minutes, what did you do next, or what happened next?
So then the blot was covered with 20 milliliters of 5% acetic acid solution, and once again incubated in the rotary shaker for also 5 minutes at 50 RPM at 20 degrees Celsius.
Okay, so this brings up a good point. So if all of the steps of a certain subsection are going to be at at the same RPM, at the same degrees Celsius, you can have, everyone can write kind of like a statement in the beginning where you can say, the blot was stained with Ponceau red using a rotary shaker at 50 RPMs at room temperature for all steps. You can start with that so that you don't have to repeat that every single time. So let's actually try to build that into this. Move this down just a bit. So the blot was stained with Ponceau red on a rotary shaker at whatever RPMs, at whatever degrees Celsius for all steps. That way you don't have to keep repeating it. And then you can probably just revise this part. We'll just erase all this and rewrite it. Okay. You can say 20 mils of the appropriate Ponceau red in acetic acid was added. And then you can simply just talk about the incubation time. Because those times might differ for each step. But if every single step was 5 minutes, you could have added 5 minutes up here too. But since the time does differ, we'll just make sure that we establish that per step. So the 20 mils of the blah, blah, blah was added. And then you also said 20 mils. Then here's your occasional then, 20 mils of the destain. So I think we just said it was acetic acid, was added for whatever number of minutes to destain the blot. And here is the maximum amount of explaining why, just to destain, just so that it's clear. And I don't even know that that's absolutely necessary, but it's just it's 2 words. You can definitely add it. You can put that in the beginning of the sentence. To destain Ponceau red, such and such was added. So now let's go to group 2. What do we talk about next? So this might be one sentence. Okay. So what happened next, group 2? Thank you, group 1. Group 2.
Okay, I'll go.
Thank you.
The data, well, once it was, the data was recorded in a photo, and then there was 2 washes of the distilled water at with the same steps.
Okay, So you are mentioning that the data was recorded, right?
Yes.
So the blot was imaged using some sort of camera. So it's still my same old good old phone, unless the video tells you otherwise. So it was imaged with what? Then destained or washed with deionized water.
Yes.
Okay, you can even say twice. Okay, then what happens? So now if you imagine what's happening here, the little white piece of paper had some red on it. We try to wash off some of the background of the extra red stain. We took a picture of it with our cell phone. Here we go. Okay, and then we put it back in the Weibo and we washed it with water or rinsed it, whatever we did. And then what do we do next after we have washed it twice with DH2O? Is that it? Arlene, we can stay with you if that was not clear.
Yes, I believe. And then it's just we stored it. Is that the one we stored in the -20 freezer and saran wrap, allowed to dry?
No, that was after we transferred it. After we
Okay,
transferred
sorry,
it.
I have that right at the end. Okay.
Yeah, so actually that's something good that we should probably add up here. After we transferred it for 350 milliamps, blah, 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 blah. You can say the blot was stored, because that's important. The blot was stored at whatever, what I think we said, -20 degrees until the next step. And one other thing that I did not hear from the class that I thought about in the break was transferring this to a nitrocellulose membrane. We need somewhere in here that the membrane, we need the membrane product name and number. So I don't know exactly, that's probably going to go somewhere in this region. So I'm going to write it in pink so we remember to add it. So the product name and the vendor and the catalog number for the nitrocellulose membrane, because we bought it from a company. We didn't make this in our lab in a certain way. Okay, great. So after you have this, then it's ready to go, right? It's ready to go into the next step, which is the immunoblotting step. So if that is it, I'm going to open it up to anyone else. Is there any other questions from groups? Because sometimes I do forget certain details. Is there anything else that you had in your notes from your group that you think need to go in this method section here? Okay, so
Sorry,
yeah, go ahead.
do we need to include the increments, like the time increments?
Of what?
The soaking.
I don't see soaking on here. So can you be more specific? Soaking of what?
So after we like do the adding the acetic acid onto the blot, and then do we soak it after, like in the acetic acid for 3 minutes?
Yeah, so it's here for you to fill out.
Oh, I see. Thank you.
You're welcome. Any other questions? But yeah, anytime you're adding something besides water, it's always important to add the concentration, the time, and the temp if those details are available to you. Okay, great. So you can see the section for Ponceau again, really, really short. We're talking like a few sentences. So when you're writing, keep that in mind that you want to avoid explaining, you want to avoid going into extreme detail. Go back to the manual and make sure that you're not missing anything and cross-reference it with the video. But I think these are the important things here. So just reading these details, I know exactly what I would do with that blot. I'd plop it in a little weigh boat. I'd add some 20 mils of Ponceau red. I know exactly what to make my Ponceau red stain with. I know that I probably need to rotate it either by hand or if I have a shaker, awesome. There's a certain number of minutes I do this, and I can read these details if they were even if they were written in past tense, I would know how to do this myself. And so, you know, I think these are the important pieces. So let's get into the predictions of what would happen. So we'll move on to group 3. So group 3, why don't you go ahead and unmute yourself and describe with descriptive words what the nitrocellulose membrane would look like if the Ponceau worked and your hypothesis was correct. So let's just talk about what we would see overall for the samples if the Ponceau worked. Group 3.
So if the Ponceau stain worked, we would see 4 columns with bands present. So the first one to the farthest left would be the marker. Next would be the sample with the 12-hour photo period. And then the next one would be no light exposure. And then the last one would be the 60-minute light exposure.
Okay. And so describe what, so 3 columns. So just like, you can see my notes, right? So it's gonna look like this.
No, there's bands.
Bands, starting where?
Starting at the with the positive.
Okay, yeah, so let's just talk about this one right here. So bands where, like at the top of the gel. So just try to, if this was a game, try to explain with words how I should draw this on a piece of paper.
So it'd be thin lines spread out.
Spread out from where to where?
From bottom to the top.
Okay. They're all equal thickness?
Yes.
Okay, what is the one for the negative control look like?
So the negative control is gonna look the same, but we predicted that there would be more bands.
More bands. Okay, I'm gonna ask questions in a second. Okay, what is this? I just want to, we'll draw what it looks like and then I'll ask questions. What about the 60?
There'd be more bands present than the negative control.
Okay, so did I draw it according to what you explained to me?
Yeah, you did.
Okay, cool. So are all of your, and you're saying that all of them are the same thickness? That's what you guys talked about in your group.
Yeah, we ran out of time about what band size would be thicker versus thinner.
Okay, so I have some questions. Why do you think that the negative control, let me change my laser pointer. Why do you think this one would have more bands and this one would have the most bands? What was your guys' rationale for that?
Because at the end of the experiment, excuse me, the 60-minute, the 60-minute light exposure had a greater concentration than the positive control.
You mean after the Bradford assay?
Correct.
Okay, so this is based on the concentrations of the positive control, the negative control in the 60 minutes. You said the positive control had the lowest concentration, the negative control had a medium concentration, and the 60 minutes had the highest concentration. But let me just remind you that this is why we did these calculations, so that when we were starting the Western blot, every single sample, you're starting with 30 micrograms. You're not starting with different amounts. So that's why we had to do this. So we actually saw that sometimes you do get different concentrations of your proteins at the very start. And you don't want that at the very start. So you do the calculations. And then when you load your Western blot, you're using 30 micrograms of the positive control, 30 micrograms of the negative control, and 30 micrograms of the 60 minutes of light control sample. So you're starting with the same amount of protein. So given that information that I just said right now, how would you re-describe those bands on this blot?
So it wouldn't have anything to do with the concentration because they're all the same.
Yeah, okay, so the way that you would change it, so let's keep the positive control the way it is. So now let's redraw the negative control. How would that look compared to the positive control?
Well, they're separated by molecular weight, correct?
Mm-hmm.
Can I phone a friend? Someone.
Yeah, of course, of course, of course. Yeah. So someone else in this group or even in the class, what do you think the negative control should look like?
Sorry, I was the one that drew the pictures and I messed up. I did it based on the results, not on the hypothesis. I misread it, sorry.
Okay, that's okay.
Yeah.
So having this conversation, and now we know that the exact same amount in theory of protein, we started with the exact same amount of protein in all of the lanes. And Ponceau, we talked about last week, stains all proteins. Okay. So how would the negative control look?
It would have less banding and like thinner bands maybe.
Why if you start with the same amount of protein?
Because I guess, or maybe it would be the same then.
Okay, anyone else want to chime in and think, so we have less bands or the same? So at this point, I'm going to ask everyone to just vote for one on the chat. Just say it will look the same as the positive control, or it would have less bands than the positive control. Go for it, just add it in. Yeah, it should look the same. In theory, it should look the same. If you started with 30 micrograms here, and you started with 30 micrograms here, and you started with 30 micrograms here, you would assume that plants make a bunch of different proteins of a bunch of different sizes. So you should have large proteins and small proteins as well. Maybe there are different amounts. So maybe the negative control makes more larger proteins. Maybe the negative control makes more smaller proteins. We don't know, right? So the idea here, and this was part of the discussion last week as well, is that you would expect bands of all shapes, not shapes, all thicknesses, but all sizes in terms of molecular weight for all three samples. That's if the Ponceau worked, because all of the plants, no matter what they were grown in, are going to be making proteins of all different functions. So, you know, even these ones here, they're fighting to survive. There are survival proteins. Maybe they're making a bunch of survival proteins because they're like, well, I can't do photosynthesis, so I'm gonna have to do something else. They're still making proteins. So this is what it would look like if the Ponceau worked. It has nothing to do with concentration, which is why we did the Bradford in the first place. Okay, so what's this group 2? Let's move on to group 3. Is there anything you'd like to add to this that?
Sorry, we were group 3, so it'd be group 4 now.
Okay, so group 4, thank you. Group 4, how could you modify this picture so that you would have evidence that your hypothesis was correct?
I guess, so then we would want to make sure that, so that there'd be the same number of banding for each of the 3 samples. It's just a varying in thickness.
Okay.
Based on the protein concentration.
Okay, but what is your hypothesis about? Like the hypothesis for the whole entire study?
Oh, that plants that undergo 12-hour photo periods will have higher rubisco concentrations. Plants that undergo 60-minute photo period will have like somewhat high, and then in absence of light will have no presence of rubisco or very little. Okay.
So kind of like this, right? Where the rubisco expression in the 60 minutes, the plants exposed to 60 minutes of light will be higher than those that were grown in the absence of light, but less than those that were grown in a 12-hour photo period, right? You agree with this symbol here? as your
Correct.
hypothesis? Okay. All right, so if that's a hypothesis, how would you know, or what would be evidence, where would you look on this plot for potential rubisco? What do you know about rubisco that might tell you, maybe this red band is rubisco? So where would you look on that plot?
Somewhere on the ladder where we do have a presence of rubisco, where we know rubisco is.
Okay, so where is that? Where do we know rubisco should be? If you're, yeah, go ahead.
Is it the 55 the KD?
Okay, so that's the size of the large subunit. What's the size of the small subunit?
I want to say 13.
Okay, great. Good job. Okay, so this molecular weight marker, let's pretend this pink band was a 50 kilodalton band. And let's pretend this lower pink band was a 15 kilodalton band. Okay. So if your, I'm gonna make this nice and big. If your hypothesis is correct, I would say you need to draw specific bands right in this region, a little bit higher than 50, because that's going to be a clue. So this would be a clue about rubisco. Remember, Ponceau stains all proteins. So maybe there's other proteins that are 50-ish kilodaltons big. But since we know rubisco is made in this giant quantity, we could probably look there and say like, okay, well, maybe these bands are rubisco. And then if this is your 15 KD, this bottom one is your 15 KD, you would also have to have some information about rubisco there. So let's erase this and draw it in so that it mimics our hypothesis. Okay. So we need something in this vicinity and the lower vicinity. So in terms of thickness, so we'll go back to this right here. Rubisco we know is composed of large subunits and small subunits. And you just finished saying, group 4 just finished saying that they hypothesized that the testable sample will have more rubisco than plants grown in the absence of light, and less rubisco than plants grown in a 12-hour photoperiod. So let's convert greater than and less than, let's convert it to thicker and thinner. So how would you describe the relationship, or how would you describe the band of the plants grown in 60 minutes of light compared to the plants grown in the darkness? Would these plants have a thicker band or a thinner band?
So it'd be a similar relationship in the sense that the 12-hour, the plants exposed to 12-hour photo period would have a thicker band,
Okay.
thicker band than the 60-minute photo period.
Okay.
And then the absence of light would have a thinner band than the 60 minutes of photo period.
Okay. True statements, all I'm going to ask you to do is switch the order so that you're comparing 60 to the controls. So if you're saying the band in 60 minutes of light will be blank compared to the negative control. So how would you describe it? So the band in the 60 minutes of light will be blank compared to the negative control. Thinner or thicker?
Thicker.
Yes. Okay. You always compare the testable sample to the control. And then we'll do it again for the positive control. Okay. So the banding pattern in the plants exposed to 60 minutes of light will be what compared to the positive control?
Thinner.
Yeah, great. So if that's the case, that's your hypothesis. So the evidence for that, you've just come up with the evidence, which is why I wrote it in red. So now you just add it to your plot. So you say this would have, it would be thicker. Oops, goodness. This would be thicker than the negative control, but it would be thinner than the positive control. And that's not just going to be true for the large subunit because Rubisco is one large entity. So that same banding pattern should also be relevant or predicted for this area down here, slightly below 15 kD, something like that. Okay. Is there questions on that? Okay, so part of the lab report asks you to make a model schematic of your predictions for the Ponceau stain. So now you have that information if this was your hypothesis, right? So, but it's really easy to change. Like if you hypothesized that the 60 minutes of light and the positive control will have approximately the same Rubisco, draw those bands equally as thick. But one thing to remember is these just provide clues about it being Rubisco. You have no right to say this really is Rubisco. It just happens to be, hey, plants that are grown in 60 minutes of light, they will have some sort of band F55 regardless. And that probably might be Rubisco, but we don't really know for sure until the next step. This just provides some clues. It's like, okay, things are looking the way that they should be looking. Now let's confirm with the immunoblock. Okay, so thank you to groups 3 and 4 who participated in helping to build this prediction. Great. So now we'll go to group 5. So we'll keep the same order of these samples, and let's just say that the positive sample worked properly and the negative sample was the sample that you forgot to add, and the 60-minute sample was, it also worked properly. So group 5, just go ahead and unmute yourself and tell me what you think the Ponceau stain would look like. So you're saying instructions to me to write, to draw this for you. So go ahead, group 5.
So we said that the red stain bands would appear thicker widths near the top of the blot and thinner width near the bottom of the.
Okay, let's go through each one the same. So if the positive sample worked the way that it should, so we just made a picture of a blot that worked. So in this second picture, we're saying the positive control worked, the 60-minute control worked. Oops, you forgot to add this. So what would the blot look like?
So they would also kind of look the same, as in, so the positive would have the thinner ones at the top and then the thicker ones in the bottom for the stains.
Okay, what about the 60-minute light?
For the 60-minute light, we said that they would have the thinner bands at the top.
Okay, so I'm gonna just have you look at this one more time. So this is a picture of Ponceau stain that worked. And so all I'm asking you to do is describe, so if this is a picture of the Ponceau stain, for the positive control, this is what it would probably look like down here. So you could just draw that same exact picture the way that we drew it up at the top. Okay. We're also saying that this worked. So if this is the picture of the Ponceau stain that worked, then this lane, or this lane right here, would very much resemble this lane up here. So draw it the same. What if you forgot to add a sample? What would the red look like in this lane?
The column would be blank.
Perfect. That's all I'm looking for. That's all I was looking for. There's nothing here. It's totally blank. Okay. Why? Why would it be blank? Why would there not be red?
Because we forgot to add the sample.
Okay, but you're not telling me why it wouldn't be red. So if we forget to add the sample, what is not there?
The bands, the red stain, because
And what is the red stain?
the proteins.
Okay. So if there's no sample, there's no proteins. If there's no proteins, there's nothing for the Ponceau to bind to. And that's if there's no Ponceau to bind to, so no Ponceau equals no red. Okay. That's why it would be blank. So if you did get something that looked like that, then you would stop your experiment and start it over. Okay. Questions on that? Okay, great. Okay, so let's go on to the last prediction. So the last prediction I asked you to do was to predict whether samples were improperly denatured. So if they were not fully denatured, meaning they're still maybe stuck in tertiary, or maybe you didn't pull all the subunits apart or anything like that. So let's say for this one, the negative control worked. Actually, all the samples, so forget it. I take it back. For all the samples, the denaturing process did not work properly. They were under denatured, not fully denatured. So group 6, please describe what the red staining would look like.
So I think the samples would kind of just be like cut off at a certain location, because
What does that mean? What does it mean, cut off?
like if some of them were like transferred correctly and some of them didn't, then I don't think the ones that didn't would be there at all.
So if you had to just tell me, if you had to explain to me how to draw this, what would you tell me to do with my pencil right now?
So I'd add the bands in the positive column, but either cut them off at the top or bottom, depending on what side was actually cut off.
Okay, so you were good until you said either the top or the bottom. So you got to pick one. So let's think about what an SDS page does. So what does an SDS page do?
Separates the proteins.
By what?
By size.
Okay, so what kind of proteins would you find at the bottom of a gel? What size proteins? Smaller
ones.
Ooh, try again.
Smaller, sorry.
Okay, so small proteins, just think about it like this big complicated web. Small ones have no problem getting through the web so that they can travel down to the bottom. Large ones stay at the top. So normally you have small proteins, so small KD number at the bottom and large KD at the top. Now let's talk about proteins being denatured. So let's say that there is a protein in quaternary structure versus its single subunits in primary structure. Which one would be bigger? The protein that's still in quaternary structure or the individual subunits?
A protein at quaternary structure.
Okay, so you would say this is larger and each of these are small, right? Small. So given this information here, if we're saying, oh, okay, so let me ask you this. So if something was denatured fully, what structure do we have them all in?
The first.
Quaternary, tertiary, secondary, or primary?
Primary.
Primary. Not denatured means what? What structure would you have or could you have?
4 or quaternary.
Yeah, well, you can have other ones too, right? You can have maybe some in secondary, tertiary, quaternary. Okay. So we will write one more thing. We already said that things that are in their primary structure are small. Things that are in their other conformations or folded conformations are large. And so we're interested in predicting what the gel would, or what the blot would look like if things were not denatured. So we know small things / denatured things would be at the bottom. Large or not fully denatured would be at the top. So now tell me how to draw the red on this plot.
So the top part would be cut off because they're not denatured, and the bottom ones would still be present because they are.
But if we did not denature them, so the samples were improperly denatured, they were not fully denatured. So I guess maybe you could be right where you have a lot of proteins up here, right? Because they're still kind of clumped together, and maybe some of your proteins did denature properly. So it might look something like that. And maybe some of them didn't denature at all. But the evidence that if you had a bunch of redness at the top of the plot and nowhere else in the gel, this really does indicate that the denaturation process was not successful. Okay. All right. So thank you to group 6. Any questions on anything that we have described here? Okay. So I really want you to go over these notes and expect to see this in an upcoming either worksheet or some kind of activity where you're asked to do something similar to this, because I think that we need to think about this a little more deeply than just talking about it today in lab. Okay, any questions on Ponceau staining, the things that you may see, why we drew things the way that we drew them, or on the methods?
I have a question.
Go ahead.
So when we got our results for the Bradford assay section, it showed that, I feel like you might have touched on this briefly during lab today, but I wasn't exactly sure. We saw that there was a higher absorbance for the absence of light samples, but we were expecting to see the opposite, like higher absorbance for the like 12-hour photo period, because they would have more proteins and thus a higher concentration.
So my question for you is, why did you expect the plants grown in a 12-hour photo period to have more proteins?
Because I think we were thinking that there would be more Rubisco present for photosynthesis, and then that would increase the overall protein concentration.
Okay, but is photosynthesis the only cell process that a plant cell does?
No, and so that's what we were thinking, that maybe there was like, it's because Rubisco hasn't been separated, like it's just protein concentration in general and not strictly Rubisco that's being, okay.
Yeah, and I mean, there's kind of like a fine line there, because we know that plants, the majority of the protein that a healthy plant makes is Rubisco. So you might say, well, if a plant's not making Rubisco, then obviously they're making much less protein. But is that true? I mean, if we're trying to survive, we're going to make a bunch of other proteins that help us survive. Like if we can't do photosynthesis, yeah, we're probably not making those photosynthetic enzymes. We're probably trying to make other enzymes that will help us somehow get cell energy so that we can survive. So you got to just remember that photosynthesis is not the only cell process that these plants are doing. Yeah, the ones that are grown in a healthy environment, yeah, the biggest cell process or the most common cell process that the cell is doing is going to be photosynthesis. But the other plants, even though they're not doing photosynthesis, they might be trying their hardest to do cell division. They might be trying to do some kind of alternate glycolysis or something so that they can survive. So I think that you need to rethink about, what does total protein mean? Because we're definitely not just looking at photosynthetic proteins.
Right. Okay, cool. Thank you.
You're welcome. Okay, great, good. Yeah, this is the time. Ask me the questions if you've been really thinking about this, and if you've been thinking critically about it. I'm happy to discuss those things now. Any other questions? Okay, so I think what I'm gonna do is I'm going to leave it up to you and your group to think about the immunoblot. So I think we have a pretty good understanding for how to write concise, succinct methods. So the methods for the immunoblot is going to resemble this very, very closely, but you're just going to use the correct reagent. So I don't think we need to use time, another breakout room, to kind of harbor in what a good method section is. I feel confident that you can write the immunoblot section for this paper, given the conversations we've had today. On the report, you also have to draw a schematic of your immunoblot predictions. So given our conversation about what we discussed up here and what would the evidence be if the immunoblot worked and your hypothesis was correct, what would your bands look like? At least you can say they're not red. For sure, I know you can say that. But what would they be? Thickness. Use this kind of thought process here. What would the thickness be? Would you see them at 55? Would you see those bands at 13? Both? You don't know. I've provided the information for you to get the correct answer in terms of which band size you should see. But I think this will be something that you and your writing team will do together. Since you and your writing team have the same hypothesis, perhaps you and your breakout roommates today did not have the same hypothesis. So I am going to stop sharing those notes and then, come on. Go to Canvas really quickly and just show you what is on the table for next week. Excuse me, let's see. Okay. Okay. So next week, do keep in mind next week the exam is on Thursday. And we're not doing an asynchronous lab for next week. However, it will be pretty short. It's more going to be a like an informal results and discussion writing workshop for the Ponceau stain and the immunoblot sections of this lab report. So what you need to do by next week, and I would get it started now, so you're going to add to your outline one more time. And this time what you're going to be adding is the remaining sections. So all that's left for this full outline are the results section for the Ponceau stain and the discussion and the figure to go with that. And then the same thing for the immunoblot. So we've done the method section, you've written it, you can revise it. We've even talked about some of the predictions that you can make, so you can make the model for these predictions. And so essentially by next week, you should have not a full written draft unless you want to make it into paragraph written form, but I think that you're a-okay leaving it in formal, detailed outline form. So your topic sentence, your bullet points, and your transition sentence. So you want to have the full outline for the Western blot study. Next week in lab, we'll talk about that. And then there are, so come with questions too. So if you've been analyzing this data, you maybe have some questions that you want to ask. I'm happy to address all of those next week in lab. And that's it. So I will get the assignment all written up into Canvas right after lab right now. And so I already did write the assignment for your notes today. So it's right here. We'll just call it week 8 participation. So only the people in the Wednesday lab have been assigned this. And so you just convert whatever notes you have right now into a PDF. And then at the top of that, please include all of your names of your teammates, that it's Wednesday, and your breakout room number. And just do this by 2:15. So you have an hour to do this. Please do not add people that were not in your breakout room, because I know who's in the breakout room. one, so please don't do that. And one person turn it in. All the others that are in the group that did not turn it in, same old, just write your teammates' names and your breakout room in the Canvas comments box and you're good to go. And it's worth 5 points. So you either get it or you don't. All right, questions? Okay, no questions.
I had a question really fast.
Yeah, sure.
Oh, sorry. I was just wondering, so just to clarify, we are not doing a section on immunoblot in these notes. That's something that we're doing with our writing group, right?
Yeah, because I think we've kind of harbored this idea, and I think it's probably more important to talk about the predictions with your actual writing team versus your breakout team. So this is something you do when you log off and, you know, do whatever you're going to do with your writing team. You're welcome. Any other questions about what was covered today? Go ahead, Greg.
I was just wondering, did you say that you already posted the results, like pictures of like the immunoblot on Canvas? Because I looked. Okay.
Yeah,
gotcha.
I'm going to do it today after class too. So after class today, what I'm going to do is write up the assignment prompt. So it's like outline 3 should contain these sections. So I'm going to do that. And then I'm also going to link in the pictures. And so what you'll see are pictures of what the plants looked like. And so some students like to make a figure for that. So what the plants look like after they were grown for whatever it was, 16, 17 days. There are pictures of the gel as it's running at different time points. That's just for you as a student to see what it would look like at different time points. That's not something that you would put in a report. There is a picture of what the membrane looked like after the transfer. So you can see that the molecular weight marker did in fact transfer. That also does not go in the report. That's just for you, maybe something that goes in a lab notebook. And then you do have your Ponceau stain and the immunoblot.
Awesome. Thank you so much.
Yeah, you're welcome. Anything else? Okay.